Hey, Bio 2 students, Mr. Yant, starting a new chapter here. So, we're ready to kind of flow into genetics. We kind of went over some, sorry about that. Kind of went over some mitosis, meiosis, which kind of gets us going in that direction of genetics. So we're just going to jump right in. So the guy in the circle there was the most famous guy in genetics, the father of genetics, named Gregor Mendel. Okay. Now, I think a big misconception is that Mendel was this monk that just kind of stumbled upon genetics, which isn't true. I want to kind of clarify that a little bit. So, when you're talking about genetics, we're talking about the science of heredity, which is really the science of potentials, how traits are passed on from, it could be to cell with mitosis and making more blood, skin, muscle, bone during your lifetime, or better yet, passing it from one generation to the next as far as egg and sperm cells are concerned. So the big thing with genetics is variation. Variations are changes that frequently occur in heritable characteristics. We all know there's variations, and we're all unique people. We have lots of different genes for the same trait, maybe blue eyes, green eyes, brown, hazel eyes, whatever. But really, when you look at genetics in the realm of biology and you really look at it in the realm of evolution, it is the building blocks of evolution. Evolution could not take place. Animals could not have changed from one look to another look over time without genetics being in the middle of all of it. So we call it Mendelism today, or Mendelian genetics. So this Gregor Mendel, again, was not this uneducated monk working in a monastery. He actually went to the University of Vienna. He studied physics and chemistry and botany and all this good stuff. And later, around the mid 1800s, he came back to the monastery and basically started his landmark genetic experiments. Okay, and here's what's amazing with Mendel, and some good videos you can watch if you want to watch them sometime. Mendel was this extremely successful. For three reasons. Let's kind of boil this down. Maybe a little Bio 1 review here for a second. So, first of all, he picked a perfect plant. He had some background in genetics. He was really good at gardening. I don't think this was an accident. If he would have picked some kind of like a snapdragon or some plant like that, he may not have came up with his conclusions. So he picked a perfect suited plant. He was really good at the scientific method. He looked at only one set of traits at a time, but he looked at different traits, but he followed them through several years of generations. And remember, Mendel did his experiments over a 10-year time span. Can you imagine that? I have a hard time getting you guys to focus on a 2-day lab, let alone 10 years. So really important that we understand that part of it. And probably the most important of all of them is number 3, and that is he was great with statistics. He was great at keeping data. And remember, long before computers, everything was, written down on paper in a journal. Pretty amazing what type of things he found. That's what we're going to talk about today. Okay. Now, when you look at Mendel's experiments, okay, so understand that Mendel's experiment, his experiment really isn't that wow, it's impressive. It's his conclusions. And it's sort of like I talk to students all the time about, it's one thing to make a graph, it's another thing to be able to interpret a graph. And he was really good at interpretation. Okay. So first of all, he took his, and some terminology for genetics, he took his parental generation, offspring, we call them P's, and he basically did a very controlled, predictable experiment. He took a bunch of them and basically set them in the corner of his garden and let them self-pollinate. Okay, and then he basically then took that population, and then that was his breeding stock, and then he decided, hey, what would happen if I crossed two different traits together? Okay, so he knew from his control group, Purple produce purple. Yellow seed produce yellow seed. Wrinkle seed produce wrinkled seed. But when he started saying, hey, what happens if I cross a purple with a yellow, or a purple with a white flower? That was where he came up with this unbelievable statistics now, or findings, I should say. So there was basically three things that kind of spun out of this. So understand, Mendel is back in the beginnings of genetics. So let's put his thinking on for a second. So let's say you have no knowledge of genetics. So you're going to cross a purple flower with a white flower, okay? And if you were Mendel, you'd probably think something in between, right? That's what he predicted, but that's not what happened. It's all purple every single time, okay? Now, he didn't know this P, little p, big P, big P. He just thought, okay, purple with white, I'll get some kind of blend, or I'll get both of them. Not true. They're all purple. Now, here's what was crazy. Then he took two purple ones. If you were Mendel, you'd probably say, okay, the second generation, I'm probably going to get more purples. But no, white showed up again. Okay. And he's like, wait a minute. How did it disappear for one generation and then all of a sudden it came back in another generation? It never disappeared. We know today, and this is what Mendel came up with, that we have some traits that are dominant and some that are recessive. If you pair up a dominant with a recessive, the dominant one shows up and prevents the other one from having an effect. Not that it disappears, because if it gets paired up with another white flower, it could reappear as it did here, only 25% of the time. But this was big, not the experiment. You're like, that's not a big deal experiment, right? It was that findings, okay? So he came up with this segregable particles of inheritance. So looking at his experiment, he was like, okay, so both parents contribute factors, he called them, we now call them genes. And it doesn't matter if these factors are expressed in the offspring or not. You get half of them from one parent, half of them from the other. And think about what this really explained. This explained, why do I have two parents that maybe have black hair? How do they have a blonde-haired child? Big deal, right? So the appearance of an individual, its phenotype, doesn't always tell you what his genotype is, what his genes are, because of this dominance and recessiveness. Now, remember that dominant and recessiveness, that can be, that's always a principle, never be a law, because there is some exceptions to that. We'll talk about that later, all right? Now, the other thing he figured out is that I could have alternative generations of, or alternative versions of maybe that one trait. So, as I said, maybe hair, black hair, blonde hair, right? Red hair. I could have blue eyes, brown eyes, green eyes, hazel eyes, right? So we call these alleles, not something Mendel came up with, but a really important concept to understand is alternative forms of a gene. And that's what basically the segregable particles of inheritance is talking about, right? Then he came up with a couple laws, right? The law of segregation, which simply states that a pair of genes will segregate or separate during the formation of gametes, egg and sperms. We now know that that's meiosis. We just had a test on that, right? But back in Mendel's day, he didn't know that. So the reason he knew that occurred is because what happened to that white gene? It obviously had to separate, and then it reappeared. That proves the law of segregation, all right? Now, you can see it here. So purple with white, all purple, but then here comes that white. And look at these numbers Mendel came up with. Crazy numbers, all right? And the other law that Mendel came up with was this one, all right? The law of independent assortment, which simply states that that there's nothing that controls that. So in other words, what fifth chromosome you get, no other chromosome pair determines which fifth one you get, or which seventh one you get, or which 14th one you get. We all know this from meiosis. They line up at the equator, the spindles pull them apart, and which one do we get? Which sperm, which egg, right? It's random. So we'll do a lab with that. We got a penny lab we're going to do, which sounds kind of weird, but it kind of proves that. So those are three big concepts that Mendel came up with. Principle of dominance recessiveness, law of segregation, law of independent assortment. And out of that came some terminology: homozygous pure, heterozygous hybrid, monohybrid, dihybrid. These are all terms we now use, okay? Homozygous imperatus means we have the same allele, either two big capitals or two lowercase, and that's that dominance recessiveness. If you remember from Bio 1, we use capital letters for dominant, lowercase for recessives. Same letter because we're looking at one trait. Heterozygous hybrid, you have different. Monohybrid, you have one trait. Dihybrid, you have two, okay? So just a little introduction to genetics. We'll kind of build on that. Hopefully that makes sense. If you have any questions, you know where to find me.